Start. So this is our today's talk, and today the topic which we will going to talk and we will try to address is a very special and like one of the personal favorite of mine as well, and that is why I don't know much about that topic either. The name of the today's topic is DNA repair. So we will be talking about the DNA damage. What are the possible reasons behind DNA damage, and how we can how our cells naturally repair that damage. Before taking you to the slide, let me try to help you to sleep well at the start. Sona nahi acha? Okay. So we have like trillions of cells in our body. Trillions. भाई एक बहुत अच्छा पाइस गाने गा रहा है. आई डोंट नो मैं उसको जाके वेल करा के आऊं कि शाबाश दे के आऊं या लैब में बंद कर रहे हैं ना तो साथ में वो गाने गा रहे हैं. So today's topic that is about the DNA repair, DNA damage and the repair. It is not the repair we are doing. It's the we will be studying the repair our cellular machinery is naturally using to fix the errors in the DNA. That's what. Okay. So all of you are there. Thank you very much. I mean, 21 students are there. Okay, that's cool. We have trillions of cells in our body, and those trillions of cells are having multiple trillions of base pairs in each and every each and every one of us is carrying that much amount of information, genetic information. That genetic information is special for several reasons. Based on whatever we have studied in the last month or so, you should be able to conclude and understand certain basic fundamentals of life, especially life in terms of molecules. So you should be able to know certain fundamental aspects of life in terms of molecules. That whatever and whoever the hell we are, we means humans or any other animals or any other species, plants, whatever and whoever the hell we are, that structure we are carrying is associated with the conservation of the genetic sequence we are carrying. So we are carrying a particular DNA sequence in us. We are carrying a particular DNA sequence. Let me hold. So every human is carrying a huge number of base pairs in every cell, and we have trillions of cells. So millions of billions of cells multiply billions of base pairs in every cell multiplied by trillions of cells we are carrying means it's an unimaginable data we are carrying as a human. Whatever the hell we are, human or any other animal or a plant, our species, our structure, our function, our integrity, our existence as human, or our existence as normal human, our existence with the functions of certain organs, the existence of those organs and the functions of those organs, the phenotype, shape and structure of the tissues present in the organs, and the arrangements of those tissues and survival of those tissues, appearance of those tissues, functions of those tissues, and are related to that information, the billions of billions of base pair of data which we are carrying as humans. If we have a mechanism, we need a very strong guarding, like a guardian-like mechanism in order to conserve not only the amount of DNA, but also to safeguard the sequence of DNA which is present in each and every of the cell we are carrying. If because of any XYZ reason, is because of any external and internal reason, if the amount of DNA we are carrying in a cell or the sequence of DNA we are carrying in the cell is disrupted, is changed, is mutated, that will going to affect the overall function of that cell. And that cell, of course, in humans and in higher animals, is a part of some tissue. And that tissue is associated in creation of a certain organ. And that organ is part of the organ system and ultimately is responsible for the very much life and survival of humans. So the change, a very small change in the DNA sequence of individual cells can be reflected, can be visible, can be seen in the overall well-being of humans. So there should be a very strong and a very effective mechanism is required, which can not only regulate, see, keep the record of the DNA sequence we are carrying, and also if something bad happens to that DNA, that mechanism should be able to respond and fix the errors or fix the mutations. The mutations we develop during our lifetime in our cells because of the external reasons, most of them, they are called as somatic mutations. I'm going to share my screen. They are called as somatic mutations. The mutations we develop over the period of time. This is called as the somatic mutation. Why I am not able to see this here? It's called as the somatic mutation. You should be able to see my screen now. I wish.
No, sir.
Not yet? Okay, I'm sharing it.
No sir, अभी नहीं show हो रही.
Okay, I think it is taking some time. I don't know why. So if oh, वो हो रही है. Okay, thank you for the feedback. Please keep giving your feedbacks in terms of reactions or using the reactions or using your voice in whatever the way you want. That will help me. मुझे भी लगता है कि आप लोग मेरे साथ जिंदा और जवेद मौजूद हैं. ऐसा नहीं लगता कि मैं.
Sir, up it's visible.
Thank you. So the DNA we are carrying, the sequence we are having, is a result of the efforts of millions of years of evolutionary process. So several million years. When evolution worked for several million years, it actually ended up in something we can call it as human. So the conservation mechanism, there should be a conservation mechanism which can safeguard the integrity of DNA we are carrying. Otherwise, whatever we are, we will not be able to live in this way. Now think about if something bad happens, to our DNA, because of the reasons I will share, what will be the reasons. If something bad happens to our DNA, that particular cell which is affected, are you getting my voice? I don't know what's going on here. Something is not right.
Yes sir, आपकी आवाज आ रही है.
I don't know why I'm not able to see myself. You people able to see me? Okay.
Yes, sir.
Okay. Thank you. I don't know. Something was not right here. So, so the DNA we are carrying is required to be controlled, is required to be conserved, because it is an effort of millions of years. So it should be conserved. So humans, the survival of humans or the existence of our species as human depends on the existence of that particular sequence. And that particular sequence is like millions of base pairs together. So we need to conserve that sequence. So there is a mechanism, and that mechanism is called as DNA damage repair mechanism. But before that thing, before DNA damage repair, we should know how many different ways are out there in which a DNA can be damaged. And what will happen if the DNA is damaged in one type of the cell, and what will happen if the DNA is damaged in another type of cells. If a damage is there in one particular type of cell, and that cell is not actually related to the very much survival of humans, or that damage can kill the cell, I can tell you this is the most I must say acceptable way of having a damage. If a damage can kill you, sorry, not kill the cell, not kill you, but kill the cell, that is the best damage you can have in your life. But if the damage is there in the cell, and the cell changes the function, it changes the function of the cell, that is something very threatening. मेरी बात समझ आ रही है? Are you getting my point? If a cell receives a damage because of the things, I will explain कि वो कैसे damage receive करता है. If a cell receives a damage and that damage kills the cell, that change in the base pairs, that mutation kills the cell, that is a most acceptable version of having a damage. But uh, if a cell receives a damage and that damage does not kill the cell, instead it changes the dynamics of the cell, it changes the functions of the cell, that is something which is threatening to the very much existence of our species. In order to understand these two things together, think about, if a skin cell is damaged because of the ultraviolet radiations, and that skin cell is dead, that will be replaced by the normal cell. But what if the skin cell is damaged because of the ultraviolet radiation, and that damage is done on the base pairs of one of the most important genes we are carrying in our cells, P53 genes. P53 is supposed to be a gene which controls the cellular proliferation, which controls the cells whether they are proliferating in a normal way or not. Now think, if the skin cell, if the skin cell is received a damage, and that damage changes the function of the P53, the P53 is no longer able to execute the function. So that cell will not be dead. That will start dividing in an uncontrolled fashion. And that skin cell, which was damaged, but damaged in a very bad way and survived, that will going to proliferate and produce multiple cells. And that's what we call as tumor. That's what we call as a tumor and cancer, or the skin cancer. So this is the very much case for the evolution to be addressed over the period of all this time. That there are always ways which can affect the sequence of base pairs we are carrying. There will always be means which can hurt the amount of DNA we are carrying, which can hurt the sequence of DNA we are carrying. So we should have a countering mechanism. We should have a response system which can address those changes, particularly in the sequence of the DNA. If you can ask me, as I shared on the earlier, as I shared with you earlier, that humans and our cells at one point of time, they do not exist as cells, particularly when they are doing the cell division. During the process of cell division, the nucleus do not exist as nucleus. The cell membrane is gone, the chromatin material is exposed to the cytoplasm, and DNA started duplicating. At that time, none of the genes we are carrying, at that time, none of the genes we are carrying are executing any function. It's all about DNA duplication. It's all about chromosomal replication at that time. So that is the time when our cells need a vigilant procedure, a vigilant proctor, which can observe the activity of DNA duplication and which can confirm that whether the new sequence of DNA is created error free. If there are a lot of errors occurs during the process of replication,
सर ये बात दोबारा रिपीट कर दें.
कौन सी वाली आयशा, कौन सी वाली, अभी कितनी पीछे से? ओके, मैं कुछ रिपीट करता हूँ.
सर वो सेल क्रिएट वाली.
Okay, so our cells, we are like carrying trillions of cells. So even if few percentage of those trillions of cells are undergoing cell division, that means those cells are not existing as cells at that time, because their nucleus is gone, nuclear membrane is gone, their DNA is exposed, and the replication machinery will start duplicating the DNA. Now once that DNA is duplicated, that is the time when the chances of incorporation of a mismatch, the chances of incorporation of a mutation is very high, because all the DNA is exposed, all the DNA is required to be created once again. So the chance of having an error during the synthesis of DNA is very high. Even the Taq polymerase, which we are using during the PCR process to synthesize DNA, it has an error potential. Not the potential is a good word. It has it can create one error like out of 100,000 base pair incorporation. It can introduce like one or two errors in that time. It seems like less, but in the context of millions of billions of base pairs we are having, it's a huge mistakes or huge number of mutations. So at that was a time when we can assume lot of DNA damage will be there, or lot of errors in the sequence of the DNA might be possible. So we need a system which can not only monitor the process of replication, but also monitor that whether the incorporation of correct base pairs are going on or not. If one of the thing, if you remember, the DNA polymerases which are used in the DNA synthesis, they are carrying one of the domain which is called exonuclease domain. That exonuclease domain is responsible for analyzing the newly incorporated base pair, and they check whether the complementation is perfect or not. If a base pair is wrongly incorporated during the synthesis, the exonuclease remove that factor. That's the one thing, one good thing which can fix most of the errors. But even after that, several other factors are incorporated. They came and they start working on the process of check and fix. Before knowing that how we can repair the DNA, the first thing we should try to see how there are many ways a DNA can be damaged. A chromosomal DNA or a nuclear DNA can be damaged because of ultraviolet radiations, the most easiest and most easily available thing. हम उससे पूछ सकते हैं फिर लोग सन बात क्यों लेते हैं? Ultraviolet radiation is like one of the very easily available choice of DNA damage out there. It can be
बट
done
सर
because of जी 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 हमजा
ओजोन लेयर की वजह से अल्ट्रावायलेट नहीं पहुंच पाती यहाँ पे.
ओजोन की
So
वजह से अल्ट्रावायलेट यहाँ पर नहीं आ रही. ठीक है,
that's why तो it's not dangerous to take a sun bath.
good शाबाश. And I can tell you one more thing since you mentioned the ozone layer. Ozone chemically reacts with the ultraviolet radiation. Okay, ozone में क्या होता है कि ozone जो है it absorbs chemically the ultraviolet radiation. The oxygen absorbs ultraviolet radiation and it will become ozone. ये एक रिवर्सिबल केमिकल रिएक्शन है. तो हमारी जमीन के इर्द गिर्द सिर्फ ऑक्सीजन है. It's oxygen only out there. But the outermost boundary of oxygen receives most ultraviolet radiations. That is why that oxygen is converted into ozone. So बहुत कम ultraviolet radiation next layer में जाएगी. The ultraviolet radiation which managed to enter into the next layer will react with oxygen and convert that oxygen into ozone. So that's why that layer will become thicker and thicker, and that is one of the way that ultraviolet radiation is not able to penetrate, is not able to reach on the planet, on the surface of the planet. But the higher places, like in the northern areas in Gilgit-Baltistan, you can receive more of the ultraviolet radiations over there. And if the thickness is less, the higher chances that you at that region and that area will going to receive more ultraviolet radiation. The skin creams which you can see in the on the faces of the cricket players, sunblocks. The sunblock is a sunblock if it contains a chemical ingredient which can chemically react with the ultraviolet radiation. It's not like physically you are stopping something to reach to your skin. No. कुछ लोग लगता है कि ये हम अपने मुंह पे फेयर एंड लवली लगा लेंगे ना तो ये भी लगेगा जैसे सनलाइट को रोक लेगी. The answer is no. अल्ट्रावायलेट रेज को रोक लेगी. Answer is no. The chemical ingredient present in the sunblock, it chemically react with the ultraviolet radiation and block it and convert it into something else. And that is why the ultraviolet radiation is not able to reach to the skin. So that is a DNA damage. This is one of the way. Radiations are direct and the most easily frequent option of creating a DNA damage. But if a DNA damage occurs in our cells, in our tissues, such change in the sequence of the DNA, such mutation in the DNA is called as somatic mutations. The mutation we acquire during our lifetime. It can be because of some toxic regions. It may be because some DNA damaging regions present in the food, in the environment, in the pollutants, in water, or in the air. But if the DNA damage occurs in a cell of the body, that is called as somatic mutation, and that can be very fatal, that can be very bad. It can be converted into cancer, or it might cause the cells to die. So that's understandable. But if a DNA damage occurs in the cells which are creating sperms and eggs, that means that error or that mutation will going to pass on to the next generation, and that will appear in as an error in the next generation. The same thing was happened with the people in the Hiroshima and Nagasaki case. जो मर गए मौके पे वो मर गए because of the physical injuries and burning and physical damages. The people who have absorbed enough radiation at that time and they able to survive after that, they were doing the things right, but they have absorbed enough radiation that their gamete producing cells, the testes or ovaries, several genes got mutated, and when they produce sperms and eggs, they are carrying the mutations, and that mutation is passed on to the next generation. And that is why you might heard the stories that even after the Hiroshima and Nagasaki, several generations were born with deformities and with errors. Okay, so ultraviolet radiation and other
Sir,
DNA damaging agents, they can push the errors, they can put the errors into the DNA. How? I will tell you. G, somebody is asking.
सर एक क्वेश्चन था ये हिरोशिमा और नागासाकी के हवाले से सर हमने प्रोसेस पढ़ा जीएनए सॉरी डीएनए और जीन रेगुलेशन का कि सम हाउ दे रेगुलेट अगर म्यूटेशन होती है आप भी बता रहे हैं ठीक है तो जब अगर उनके केस में देखें हिरोशिमा और नागासाकी के केस में तो उनके जो स्पर्म प्रोड्यूसिंग सेल्स हैं फॉर एग्जांपल सम ऑफ देम गॉट म्यूटेटेड ठीक है लेकिन बाकी तो नहीं होंगे ना तो जो न्यू स्पर्म्स बनेंगे वो तो आई थिंक नॉर्मल ही बनेंगे
No. What I am telling is that errors in the cells which are producing the sperms. Okay, I am not talking about
ओके
the errors in the sperms. Agar error sperms mein hai, to they will be gone in one cycle.
यस यस
Next
सर
day they should be fine. It's not about the error in the eggs. It's the error in the cells which are generating the eggs. वही गड़बड़ है। तो अगर कोई आदमी अल्ट्रावायलेट रेडिएशन के पास काम करता है, एक्सरेज के पास काम करता है, जो which are DNA damaging. So if that damage is occur in the skin cells or in the other tissue cells, that's a little or smaller, comparatively smaller damage. But if that X-rays or the ultraviolet radiation influence and affect the gamete producing cells, that might be very toxic. उसको पता भी नहीं लगेगा ना, might have no idea कि मेरे पास क्या error चल रहा है. So क्या
यस सर
गलतियां हो सकती हैं? क्या गलतियां हो सकती हैं? पहली गलती डीएनए की कौन कौन सी इंसर्ट्स हो सकती है? डीप्यूरिनेशन हो सकती है अब डीएनए में प्यूरिन रिमूव हो सकता है. डीएमिनेशन हो सकती है ऑफ साइटोसिन टू यूरासिल. इफ साइटोसिन में से अमीनो ग्रुप निकाल दें, केमिकल स्ट्रक्चर अगर आपको याद हो, इट विल बी कन्वर्टेड इनटू यूरासिल. दैट्स अ डिफरेंट थिंग. pyrimidine dimerization हो सकती है. pyrimidine has a chemical tendency to become dimerized with each other, and these things must be repaired with the enzymes which are part of the DNA repair system. The most easiest way to deal with the DNA damage, सबसे आसान तरीका डीएनए डैमेज को फेस करने का पता क्या है? Ask the cells to stop cell division. When the cells are not dividing, they are supposed to enter into a phase called as G not phase, not dividing phase. So the most easiest thing is that you can push the cells not to cell divide, not to divide at all, to stall the cell division, or to stall the cells in one phase called G1 phase until the error is repaired. सबसे आसान तरीका ये है, भी error fix हो जाएगा तो चले जाना, cell cycle में नहीं होगा तो नहीं. तो first thing ये हमारी body का करती है, after the DNA damage, it push the cells not to go for the cell division. Wonderful response है, so that the error should not be passed on to the next generation. Error रहे तो इथे रहे. The system that detect those जी शाबाश.
सर जब डीएनए डैमेज जिस तरह सोमैटिक सेल्स में हो रही होती है तो अभी आपने ऐसा कहा है कि लाइफ लॉन्ग प्रोसेस है, तो फिर इसका मतलब है कि बीच में ये G1 फेज में नहीं आते सेल्स अगर इफ इट इज अ लाइफ लॉन्ग और हेरिडिटरी प्रोसेस.
No. At the time, the cell which is experiencing a DNA damage, the first response of the cell to the DNA damage is to stop that cell to not to go for the cell division. वो divide नहीं होएगा until unless the error is removed. जब तक DNA का error fixed नहीं हो जाए. Once the error is fixed, the stalled condition is restored and the cell can proceed further in a normal way of cell division. तो हर
तो
cells
रेडियो
अलग-अलग
एक्टिव
stage.
सेल्स, तो जो रेडियो एक्टिव तरीके से म्यूटेट हुए हैं सेल्स वो जो हैं स्टॉल नहीं कर रहे.
Now, if a cell is mutated because of radiation, that radiation can cause irreversible damages. There are several chemical reactions or errors which our enzymes can fix. I said depurination, deamination, pyrimidine dimerization. आप ये process देखना, our cells can fix them, our enzymes can fix them. But if something very weird happened, अगर हड्डी टूट गई बड़ी अजीबोगरीब तरीके से, something is destroyed without any shape and structure, It cannot be fixed. तो सेल्स क्या करते हैं, सेल्स उससे जान छुड़ा लेते हैं, उस बेस पेयर से जान छुड़ा लेते हैं, उस पोर्शन से जान छुड़ा लेते हैं, and that can leave a mutation over there, and that mutation might change the function of a gene, and that mutation might be nothing. There are certain important machineries over there, certain protein complexes which sense the DNA damage and which respond to the DNA damage. Sense करते हैं and they respond to the DNA. They are very important members of our cellular machinery. They are multi-protein complex. Most famous, you might remember, BRCA associated proteins. They are our genome surveillance complex, the BRCA proteins. They check and see whether a DNA damage is there on the gene or not, on the genome or not. They also include another very famous protein called ataxia or ATM. Do not relate it with the government of Pakistan Tehreek Insaf. Ataxia telangi actasia mutated protein, or shortly ATM. These two together not only sense but also fix the errors. How they will going to do the things, we will see after the break. Okay, see me in the next